Before modern genetics existed, scientists had no clear way to explain how traits were passed from parents to offspring. Some believed traits were blended together, while others had no real scientific explanation at all. Gregor Mendel changed that by designing careful, controlled experiments using pea plants. Instead of guessing, he counted, recorded, and analyzed thousands of results to uncover hidden patterns of inheritance. These experiments became the foundation for how we understand heredity today and helped shape the field of genetics as a real scientific discipline.
I will be talking about Gregor Mendel and his background. He was born in July 20th in 1822. His family status, he was poor and he had lived on their farms. His parents' name was Anton and Rosary Mendel. During his early life, he grew up working on a farm surrounded by plants and animals, and that is how he sparked his curiosity when it came to the pea plants. His interest was developed curiosity about nature and heredity. Education, he excelled in school, then later studied science and math at the University of Vienna. Some traits about him: he was very curious, patient, and thoughtful throughout his studies about the pea plants till when it came to the heredity part. And these qualities has shaped his future discoveries throughout later in his life.
Gregor Mendel conducted his famous pea plant experiments between 1856 and 1863 using P. sensitivum, or pea plants. He chose peas because they had clear, visible traits and could easily self-pollinate or be cross-pollinated. Out of 34 varieties, Mendel selected 22 purebred lines that consistently showed the same traits. He focused on 7 contrasting traits, such as yellow versus green seeds or round versus wrinkled seeds. Mendel crossed plants with opposite traits, like round seeds and wrinkled seeds. In the first generation, called the F1 generation, all offspring showed only the dominant trait. When those plants self-pollinated, the second generation, or F2, showed both traits again in a predictive 3-to-1 ratio, or dominant to recessive. He also tracked 2 traits at the same time using dihybrid crosses and discovered a 9-to-3 to 3-to-1 ratio, showing that traits are inherited independently. After growing about 28,000 plants and carefully analyzing the data, Mendel discovered the law of segregation and the law of independent assortment, forming the foundation of modern genetics.
Now we can talk about the contributions that Gregor Mendel made. Gregor Mendel is accredited as the founder of modern-day genetics. He laid the groundwork for the principles of inheritance. This includes the law of segregation, the law of dominance, and the law of independent assortment. The law of segregation says that each individual possesses 2 alleles and only 1 allele is passed on to the offspring. The law of dominance says that some alleles are dominant and others are recessive. The law of independent assortment says that genes for different traits segregate independently during gamete formation. A quick fun fact that I have about Gregor Mendel is during his lifetime, his findings were not popular. His contributions were not appreciated until post-mortem.
In conclusion, it's pretty safe to say that Gregor Mendel has earned his right to be talked about in classrooms all around the world, in high schools, colleges, and even middle schools. Despite coming from a poor family, he excelled in school and went on to make some of the most important scientific discoveries in his field, being titled the grandfather of modern-day genetics. He discovered the law of segregation, the law of dominance, and the law of independent assortment, and also laid down the foundation for inheritance experiments as a whole with his scientific research methods, results, and theories. Thanks to his crosses with pea plants and bees, fun fact, he has laid the ground for modern and future scientists to continue making discoveries that positively impact the world and mankind as a whole.
Mendel's work showed that inheritance follows clear, predictable rules rather than chance. Although his discoveries were not widely recognized during his lifetime, they later transformed biology and medicine. Today, the same basic principles from his pea plant experiments are still used to understand genetic diseases, inherited traits, and how DNA works inside living organisms. His simple but powerful experiments proved that careful observation and data can completely change how we understand life. Sources for this video include Encyclopedia Britannica, Mendelovo Museum, CSHL DNA Learning Center, Molecular Plant, and softschools.com.